Alright guys, so this video is going to be to help you fill out your diagram. For this particular set of essays, you have 3 diagrams that you've got to fill out. One is over DNA replication, and then 2 are over protein synthesis, and we'll do those at a later time. So today in lab, you should have done an activity where you model DNA replication. And so this first essay is going to be essentially you filling out a diagram that was represented by that activity. And so in your study guide, you should have a paper that looks like this. So as you watch this video, you may want to have this handy. And just to kind of give you a heads up, this diagram is only going to be on the test. It is not going to be on a quiz or anything like that. The quiz is just going to be over note material. And on the test, you will have all 3 diagrams, and you will have a word bank. You'll have one word bank that is containing all the words for all 3 diagrams. So just know that you aren't going to have to recall everything. You're just going to have to know where it goes. Now, I will tell you that memorization can be used here, but it's a lot easier to fill it out if you actually understand why you're putting things in the blanks. If you understand, then it's not going to be as difficult to memorize. So as we go through, we're going to kind of just review this process of DNA replication. So on your diagram, you have the picture of the DNA strand that has been opened up. Remember that DNA helicase does that. And then it's showing us that the direction of the replication, the overall direction, is going up here. And that's important when you're trying to determine the leading and the lagging strand. So essentially this right here represents the replication fork, okay? So what we're going to do is you've got blanks on your diagram. We're going to go through and fill all of them out. Now, the first thing that we want to fill out are our directions for our strands. Now, on the original strand, we had 5' at the top, okay? Which means 3' is at the bottom. And honestly, you need to know the 5 and 3 because there's a lot of blanks that are coming from 5s and 3s. So if you get those backwards, it's going to be really easy to lose a lot of points on your essay. So on the original strand, 5' was up, 3' was down. And what that means on the other strand is that it's going to be opposite. So we have 3' and 5' there, okay? Now, this black line is representing our original DNA. And so you have a blank over here on the left, and that blank is going to be the template strand. So this white piece of DNA paper was our template strand. Remember that each strand is going to serve as a template for building our new strands. Our new strands were built on the inside of our DNA molecule. So as we go through, we're going to be building a new strand here, okay? And we're going to be building a new strand over here. Remember that one of those strands is what we call the leading strand, and one of those strands is what we call a lagging strand. So this one over here on the left is being built toward the replication fork, because remember our new strands are going to be built 5 to 3. So on my new strand, 5' is on the bottom, and 3' is going to be on top. On the other side, 5' is going to be on top. And I'm actually going to write this twice over here because I'm building it in fragments. So I'm going to have 5 to 3, and then I'm going to write that again, 5 to 3. So that should take care of all of the little small squares that are on the outside and on the inside of your DNA strands. So this one over here on the left is being built 5 to 3. It's being built toward the replication fork, and it's going to be built in one long continuous strand, okay? So this is the one that we're going to call the leading strand. Sorry for my writing, it's kind of hard to write sideways. And on the other side, you notice that this one's going to be built in pieces, and so this is going to be called our lagging strand. Alright, so now we have a few more blanks that we need to fill in, okay? So down here at the bottom, I have a couple of U sequences. If it's U's, we know that that's RNA. And over here on this side, I have AU and AU. And when you did the activity, before you were able to build DNA, you had to put in RNA primers, and this is going to be accomplished by an enzyme called primase. And so down at the bottom, you have a blank square, and it's got an arrow pointing to this, it's got an arrow pointing to that, and it's got an arrow pointing to that, okay? And so these are going to be my RNA primers. So once again, you kind of had 3 arrows going like that. All right, so over here, you have a couple of blanks that we need to fill out. And so once the primers are put in, then that's when DNA polymerase comes in, and on the leading strand, it's going to build off of that primer, and it's going to add all of your DNA nucleotides based on the complement system. Over here on this side, remember, we're building away from the replication fork. And so if you did this in the activity correctly, you would have built a strand off of that primer, and then you would have come up here and built a strand off of that primer. And so over here on the side, you have a kind of like a big blank, and on the word bank, you'll have the whole phrase. You won't just have the single word, you'll have the phrases as well. So it's going to be kind of hard for me to fit over here, but what you're putting in that blank is DNA polymerase builds Okazaki fragments. And remember that on the lagging strand, because we're building it in pieces, because we're moving away from the replication fork, we have multiple primers, and we're going to have multiple fragments, and we call those Okazaki fragments. Okay, so that's what's going in the big blank over there. Now, once you built your new DNAs, remember we don't want RNA in our DNA. And so we have enzymes called nucleases, and their job is to cut out mismatches. Now, another thing that they can do is they cut out those RNA primers. So the only reason the primers get put in is because DNA polymerase has to have something to build off of. And so once the DNA is put in, nucleases, which are another type of enzyme, will come in and they will cut out the RNA primers. So in your activity yesterday, you were supposed to take out the green bases and you were supposed to replace them with blue, okay? And so even though we don't necessarily see this in the diagram, over on the side you have a blank underneath DNA polymerase builds Okazaki fragments, and it's pointing to the primers, okay? So this technically is representing what we would do after the DNA is built. And so it's going to be RNA primers are replaced by DNA. So RNA primers replaced by DNA. And that's what's going to go in that blank, okay? Now at this point we filled out every single blank except for one, and this is going to be a really easy one for you to miss, so just make sure you pay attention to this. On your diagram, on the lagging strand, right above this primer, you have a little dot. It literally looks like a hole punch, okay? There's a little circle, a white circle, and this is going to be one of our blanks, okay? Now this blank is representing the enzyme that comes in and it glues our fragments together. So we would not want our DNA to be fragmented. And so on the lagging strand and in places where we replace the primers, the enzyme ligase will come in and glue the fragments together. So that little tiny dot, and you don't have to write it in the dot. If you want to draw an arrow out to the side, that's fine. Just be sure you don't forget that blank. But in that dot, you are writing the word ligase. And that should wrap up our whole diagram. Okay, so I hope this, number one, helps to explain the activity that you did. And number two, it also prepares you for the essay that you need to do. Okay, as I mentioned earlier, if you understand the process of replication, this is going to be a lot easier to fill out. If you just memorize it, and let's say you get 5 and 3 wrong, it's probably going to lead to a lot of missed points. So just make sure that as you study it, you understand why you're putting that in the blank, and that way you're studying for both the multiple choice questions and you're also studying for the essay. So that's going to be all for tonight. I will hopefully see you soon, and I will definitely see you in videos soon for protein synthesis. Y'all have a good day.